I am the Tofu Golem. Please enjoy the video.
You begin by verifying prerequisites and then open Arlequin with your project file. This gets you into the working environment immediately. Once done, load the primary ARP dataset and the associated metadata so Arlequin can map samples to populations. Make sure to set correct encoding and checkmarker types now, because those choices affect downstream calculations. You'll see panels for data, projects, and output. Use them to confirm samples loaded correctly before moving to configure analyses. You now define which analyses Arlequin will run and why each matters to your question. Choose F-statistics or AMUVA to partition variance, exact tests for significance, and mismatch distributions for demographic inference. This selection guides computational cost. Next, set simulation details like number of permutations and bootstrap replicates to balance precision and runtime. Finally, tailor population models, group hierarchies or migration matrices, and handle missing data. Since those model choices change result interpretation, and you execute the configured runs and watch the progress indicators so you catch errors early. Once finished, open the generated output files and inspect summary tables and HTML reports to interpret FSD, AMUVA percentages, and p-values. Export tables or graphs for downstream plotting and document parameter choices for reproducibility. If results look off, rerun with adjusted permutations or corrected input formatting. One key troubleshooting tip: check for inconsistent sample labels or encoded.
Thanks for watching. Consider subscribing if we helped you today.